This is a Tedoex Media House production.
Okay, so let's start with why has this virus been so elusive to develop a treatment for? Maybe, the structure or the chemistry or the way it infects cells.
You know, I'm wondering if it has been that elusive because things have been going at such an accelerated speed. One of my worries is that people think that this is normal to slow when actually it's floored, like vaccine development, for example. I was reading this morning that AstraZeneca is saying that, that they're hoping to get readouts of their phase 3 stuff in August and September. They're already manufacturing the vaccine just on a risk basis in the hopes that it works. And their CEO, Pascal Soriot, said, if everything goes well, we could roll out in October. And that is insanely fast.
Especially because they just started like in March.
They just started. So one of my worries is that, I'll be very happy if they get a working vaccine by October, of course, that would be great. But one of my worries is that people will get the idea that, well,
this
okay, that's how it works. Yeah. Pandemic in March, vaccine in October, and anything short of that will be seen as some kind of evil conspiracy. So I actually think that things have been moving as fast as they can possibly move. And of course, that's not as fast as people want. People want a cure right now.
Yeah.
And I understand that. I want a cure right now too, but folks get tired. It's like we were just talking about with cancer. Folks get tired of hearing over and over, well, it's really complicated, but it's really complicated.
So maybe let's give a brief overview of how the whole drug discovery process works. From designing new drugs or repurposing of drugs to treat new diseases that come up.
Yeah. Well, in the context, I'll do it in the context of the coronavirus because the same lessons are in there. You've got a couple of approaches, like when a new virus like this starts infecting people. The first thing that people tried, and it's a good idea, is to see, are there any existing drugs that will do anything? Now, the odds of that happening are unfortunately pretty small, because to have an existing drug that will come in and do something totally new to a new virus is really, really a long shot. It's like going into an auto parts store and just grabbing stuff off the shelf and throwing it out at your car and hoping that one of those things will fit your car and make it run again. It's not impossible, but it's not going to be easy. So the reason people do it, though, is because these drugs are already approved. They're already on the market, they're on the shelves, and they're ready to go into human beings. And we'll get back around to why that's such a huge step. But if you find something in this repurposing, you can start giving it to people immediately. So that's why the first screens that were done against this coronavirus were all of the existing drugs. Makes sense. Now, what we saw is people grabbing at anything like hydroxychloroquine, which we'll probably get back to.
Yeah.
Or they're grabbing at all of the existing antivirals. So you try everything that's ever worked against a virus. Makes sense. But of course, every virus has got its own set of targets, its own set of enzymes and proteins that the drugs are targeting, and they're going to look different from every other virus. So you notice that, for example, the viral protease inhibitors that work so well against HIV don't do anything to the coronavirus.
Yes.
Different proteins, different fit. They're just not even recognized.
And this goes back to even testing. I remember
Sure.
someone proposed, why, if you tested for flu, could you pick up coronavirus? And I was like, no, that's not how it works.
Not how it works.
You know, it's pretty
Exactly.
specific.
Exactly. So the first round of testing was all against existing drugs. Now, as far as drug discovery, if you're trying to discover something new, oh man, you have to prepare yourself for kind of a long road there, because if you want, I'll give a brief overview of how, how these other drugs were found and how you find one for this. So first thing you need is a target.
Yes.
Well, let me back up. There's two ways to do it. One of them is where the first thing you need is a target. And with that, you look at something like the virus and you say, okay, how does this virus work? Fortunately, viruses don't have many moving parts. They're pretty stripped down. I mean, as far as I'm concerned, viruses are not even alive. They're just little bits of protein that manage to hijack everything else we've got to make more little bits of protein. So you look at that and you say, what parts does the virus have? And for the coronavirus, you can do that. You can look at its genetic code and see what proteins those code for and say, okay, and the coronavirus has 27 proteins. I think that's the right number.
Yeah.
And that's it. I mean, we have God knows how many hundreds of thousands of proteins, but the coronavirus has 27, and that's it. And one of the big ones, one of the first things it needs to work is called MPRO, the main protease. So that would be a place where you'd want to find a new drug. So how do you do that? So, all right, here's the main protease. We want to throw something into its machinery, into its active site, to gum that up and make it unable to do its job. Well, first thing you do is you get a lot of the main protease together. And there's ways to do that. You engineer other cells to make main protease for you, protein production and purification. You make a bunch of that and you start setting up drug assays. You can engineer things so that you can set up a whole bunch of compounds. And by a whole bunch, I mean hundreds of thousands, millions of compounds to test one by one against the main protease and see if you stopped it working. Now you try to set that up so the assay that you're running can maybe give off a little flash of light when you've killed it.
Yeah.
Or maybe it's giving off light all the time and it stops giving off light when you've killed it. That's because we have machines that can measure those flashes of fluorescent light really, really fast. And if you're going to be testing like 2 million compounds, you want something that works really, really fast.
Yeah.
So there's several ways to do it, but that's one, these fluorescent assays. You set up an assay like that, though, and you make sure it works, and then you run a couple of million compounds past the main protease, and you find the ones that seem to stop it working. Then you do it again at different concentrations, and you try to make sure that's real, because some of those are going to be wrong. False positives, you know what I'm talking about. You need to go back and check. So you need to find the ones that actually work, and they work when you change the concentration. You can say, aha, this one works. It cuts down the main protease by 50% when you get this much of it. And IC50, we call it. So you want a bunch of compounds that work reproducibly and do it by a way that you can actually use for a drug, because there's a lot of ways to stop an enzyme, and some of those ways will stop every other protein you give them to, and that's not going to be so useful as a drug.
Yeah.
But you'll end up with hits, screening hits, maybe a few dozen, maybe a few hundred. And then it's time for the medicinal chemists, which has always been my job, to sit down and look at them and say, okay, what do we do with these things? Do we have any other compounds, or can I buy any other compounds, or can I make any other compounds that look kind of like these? And you're looking for something called an SAR, structure-activity relationship, as you know. And that's where drug discovery really gets going, because at that point, the medicinal chemists sit down, they make hundreds or even thousands of new compounds in a whole series, like, all right, this worked. What if we make this part longer? What if we cut this part off? What if we make this part more electron rich, or this part greasier? What if we have a hydroxy on here, or a fluorine on here, or a chlorine, or a methyl, and you make all those different analogs one by one, using the organic chemistry, the organic synthesis that you learned in grad school, and you test them. And the assay comes back and said, that one was worse. That one was better. That one stayed the same. And you're like, okay, what does that mean? Maybe we need to make this part even longer. Whoop, that didn't work. Let's make sure not to do that again. And you keep going around this cycle again and again, hundreds or thousands of times, until you've got something better. So what do I mean by better? That means something that's better at shutting down the enzyme, and also something that's better at getting into cells, because something that just works on the enzyme when it's sitting in a tube might or might not get into living cells.
Especially because the cell machine, it's so complicated. There's so many layers in the body, and trying to get through and maneuver, just looking for that specific target, it's really challenging.
Oh yeah, the things you're trying to optimize for getting into a real living creature like us include, does it get into cells? Another one is, does it get out of the stomach? If you take it as an oral pill, does it even get absorbed through the stomach and intestine? Because some things don't. If it gets absorbed into the blood, how long does it last in the blood? Because some things just get cleared out immediately.
Yeah.
We call that, all that stuff, ADME, absorption, distribution, metabolism, and excretion. And some things just can't take it.
Yeah.
So you're trying to find a compound that inhibits the target, can be taken as a pill, lasts long enough in the blood where you don't have to take it 9 times a day or something like that. And it doesn't do anything else horrible, toxicology, because maybe this wonder compound you've got also shuts down an enzyme in the liver. Well, that's not good. You don't want to make people sicker when you're trying to cure them of coronavirus. So it's just like you're saying, there's like dozens of different factors you're trying to fix at the same time. Sometimes you can't fix one without messing up another. It just seems like they're tied together. You can't do it. So then you have to go back and use a different compound series and say, okay, you know what? We can't optimize that one. We're stuck. Go back. And that adds another 6 months or a year to your process.
And the teams that are working on this, they're so dynamic. You know, you have chemists, you have biologists, you have pharmacologists, you know, everyone's coming with a bit of their own expertise. And
yeah. It really takes a big, long list of people, each with their own specialty.
And this before they even put it in animals or human beings on the market.
I mean, what I do is I make new compounds, and I think up ideas to make new compounds. I do not dose the rats. They will not let me near the rats. They don't want me near the rats. I'll mess things up. Meanwhile, those fellows who are dosing the rats, or the ones who are figuring out the formulation to dose the rats, or the ones who are running the assays against the protein, I don't want them in my chemistry lab. They'll blow things up. They'll set things on fire.
That's not true. This is why I was working in assay development, and the chemists would be absolutely mortified when I walked into the lab, and I was like, I can do it too.
Exactly. And in the same way, you didn't want them coming in and telling you, hey, you know, have you ever tried running an assay like this? You're like, oh, please go away.
Let's go back to hydroxychloroquine, because firstly, I can't believe that this president keeps hammering down unproven data. But I think this goes back to some existing drugs. They can have some activity, or they can seem to target the specific proteins that we want. But then we have all these other side effects, right? Because they're not that selective, which becomes extremely dangerous because it works, but it shuts down a lot of other things. So it's not good.
It's not, And with hydroxychloroquine, that other stuff is something that It's a cardiac side effect, and it's fairly common in a lot of old drugs because back then we didn't know how to look for that.
Oh,
It's
yes.
a, yeah, QT prolongation. If you want to sound like you know about drug discovery, talk about QT prolongation. But now we know how to look for that, and a lot of drugs get stopped in development because we find that happening early on before we even go into animals, and we say, okay, got to fix that. But hydroxychloroquine will do it. A lot of old drugs will.
Hmm.
And people are like, well, they've been giving hydroxychloroquine as a preventive of malaria, or people with arthritis or lupus take it. But those people do not have the coronavirus. So giving it to someone who's already extremely sick with the coronavirus is a much different thing. And it looks like it really is more harm than good.
I think something that hasn't, it's been challenging explaining, especially how we now have new technology with things like proteomics, where
Yeah.
we can pick up all these other off targets, we call them,
Yes.
which can signal what are the side effects we would expect to see if we were to move forward with this drug, is also explaining how it's not intentional that some of this medication was put on the market.
Right. Right.
That people just disregarded all the side effects. It was just that there was no technology to do these
Right.
measurements, and that's why we
Right.
recall some of these medicines.
And exactly. And even now, with our much better way of checking all these proteins, even now we get surprised, of
Yeah.
course. And that's an important point you're making because every drug has side effects. Every single drug. And the question is, are the side effects going to be common enough or bad enough to cause trouble? But every single drug. And the other thing to remember, of course, is the one, and this is a saying that goes back to the 1500s, a man named Paracelsus. He said, the dose makes the poison, which means that if you take enough of any drug, it will be poisonous.
Yes.
Now, enough might be the entire bottle. And that's good because that means that you're not going to poison yourself, most likely. Or in other cases, like with insulin, insulin is a vital drug. If you're diabetic, it's going to keep you alive. But let's put it this way, if you take twice as much ibuprofen as you're supposed to, not much will happen. If you take twice as much insulin as you're supposed to, you're going to be on the floor. So there's a lot of different, what we call windows there, and some of them have very tiny windows.
Yeah. And I also wanted to mention, what was it? Let me just remember.
Sure.
Oh, yes. Sometimes I think as scientists, we keep finding out new things. It's the case of thalidomide,
Oh,
how
yeah.
it looks very safe in animals, but then there could be just one tiny difference between
Yes.
animals and like mice and human beings, and that makes all the difference that we cannot put
Yes.
this drug.
Thalidomide is a really good example because, of course, that was originally given to people back in the late 50s, early 60s, and it turned out to cause horrible birth defects. And it turns out it doesn't even do that in all the animal models. It's different in mice versus rats versus rabbits, total versus people. It does totally different things in those four species. So, yeah, I mean, honestly, this is not a very good advertisement for our profession, but every time a new drug comes on the market, you're holding your fingers, you're holding your breath and crossing your fingers
Yeah.
because you can test a lot, but you don't really know what's going to happen until it goes into people. That's what we're getting with the coronavirus right now. We're taking the antibodies and vaccines into people, and everyone who knows what they're doing is holding their breath, hoping it works because there's no way of knowing until it really gets into a person.
And the more experiments you run, the more answers you get, and the more your hypothesis changes.
Yeah, exactly. So in the end, it all comes down to dosing human patients. And of course, that's a good thing to lead into the clinical trials because
Mhm.
phase 1 clinical trials, you're dosing people who don't have the disease. normal patients, or as normal as you can be and take an experimental drug, but normal patients who don't have the disease, and you just give them small amounts to make sure, one, that nothing crazy is happening, and two, that the blood levels of the drug are what you thought they would be based on your animal studies. But then if that's okay, you go into phase 2, where people who have the disease are finally going to take this drug. That's the first time that happens. And then you hold your breath and hope that it worked. You know what? Most of the time it doesn't. I think one of the other big things that people don't understand about drug development is that from a compound going into humans to getting approved and going on the market, the failure rate is 90%. 90% of things never make it out of clinical trials. And a lot of them, a few of them wipe out in phase 1, but we mostly get that right. A lot of them wipe out in phase 2 because they don't do anything for the disease. Our whole idea about how to fix the disease was wrong. And then a bunch of the others wipe out in phase 3 because either it turns out that it works, but not well enough to do anyone any good in the real world, or there's some weird side effects or toxic problem that started to show up when we dosed more people, and that's all for that. Go back and do something else.
So maybe let's discuss, right now there's so many different vaccines and treatments that people are developing. So what are sort of the front runners and how do they work?
Yeah, these things are in clinical trials right now. So there's two broad categories of vaccine. All of them are trying to do the same thing. All of them are trying to make your body's immune system respond and develop antibodies and T cells that remember what happened, so that if you do get infected with the actual coronavirus, they're ready, they're prepared, they'll go in and kill it. So they're all trying to do that. But the two ways you can do that, one of them is you give people a piece of the coronavirus, like just a chunk of the outside, a piece of that protein, inject them with that and let the body react to it. Because the immune system will see this foreign protein going around your blood and go, whoop, 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 intruder alert, and start developing antibodies and T cells against it. So that's one way of doing it. And you can give a little piece of protein. You can give a virus that's been totally killed and can't reproduce, but just has all the pieces hanging off of it. Or you can give something they call a virus-like particle, several of these stuck together. Those are one way. The other way is to give something that will go in and kind of infect your cells and make them make the foreign protein. Now, that's what a real virus does. The real virus goes in, attacks your cells, hijacks its protein machinery, and forces it to make more virus. So the other way of doing this is to give people something like DNA or RNA. One way, the cells take that up and they translate it into protein. Another way is to pick a totally different virus. And there's 2 or 3 of these going right now, where you take another type of virus, and a common one's called adenovirus. You take its instructions out, because its instructions are make more adenovirus. You don't want that. You rip that out of there and you put in a little bit of DNA or RNA to make some coronavirus protein, and you let that infect people. And the final way of doing it is to actually make a weak form of the coronavirus. You can take these things and run them through cells thousands and thousands of times. And sometimes when that happens, the virus gets weaker and weaker and weaker. If you can engineer a strain like that, you can give that to people, and it won't give them COVID-19, but it will set off the immune system. So you got 2 things.
One of the type of vaccines that we also get with the shots, right? When we're growing up.
Yeah, exactly. Because the kinds of viruses the kinds of vaccines that we get when we're growing up fall into all of these categories. Some of them are bits of viral protein. Some of them are live virus that's just weakened. And Now, there are actually no DNA or RNA vaccines yet. That's a totally new technology that we're just getting a chance to try out with the coronavirus. And isn't that exciting? But we get killed virus vaccines, we get weakened virus vaccines, and we get chunks of virus vaccines. All of those are given to people now.
So, so the mRNA one is the one that Moderna is
That's Moderna.
developing right now?
Exactly. And that's a technology that's been coming on for some years now. People have been trying to get that kind of vaccine to work, and it's shown some promise, but it's never gotten as far as being dosed into people until now. So it's just happened to come along at the same time that we really need a lot of new vaccines. So this is going to be a pretty dramatic one. We're going in there with a technology that's never been tried on people to try to fix a virus that people have never gotten. So this is a bit like a movie.
So what about the people who do have COVID-19? I know one of the treatment that's available is it Gilead's remdesivir?
Mm-hmm.
So what is the technology there, and what else have people been developing?
Yeah, remdesivir has been around for a while because it was originally developed against Ebola, against the earlier Ebola waves in the DRC, in the Congo. And it didn't work that well against Ebola, unfortunately. But then when SARS came around in the early 2000s, another coronavirus, they tried it against that, and it did have some activity. The reason you're trying it against all these things is that remdesivir, in theory, should mess up any RNA virus. And all these we're talking about, and the current one, are RNA viruses. It goes after the enzyme that the virus uses to build new RNA. Problem is, it doesn't do it that well. It's okay, kinda. And that's what we're seeing with remdesivir in the clinic. It does help, but it's not a cure. It's not a cure. It will slow the virus down a bit. And compared to nothing, that's pretty good. But compared to a cure, it's not much.
Pretty far away.
But since it seems to have a good safety profile, it doesn't do that QT prolongation that we were talking about, it doesn't have cardiac side effects and all. It seems to be pretty safe, and it does slow down the virus, and it does seem to keep people from getting into as bad a problem as they get. If the virus just, lets it rip, then it's what we have. There are 2 or 3 other drugs that are coming along, that repurposed drugs like this, that might actually do as well or better. But that's the main thing we can hope for those, is that they will help some.
But they won't change.
They won't be a cure. But it's, if we can get fewer people going into the ICU, or if we can get people to die at a lower rate once they're in the ICU, this is good. This is definitely good. But in the long run, it's going to be something like a vaccine that really pulls us out of this.
So from some of the, I guess, the literature that's coming out of a couple of labs that are researching this, it seems that the coronavirus will infect the respiratory system, but then the effects are beyond just the respiratory system, with inflammation, and it's shutting down the whole immune system, which makes it much more challenging just to go after preventing further infection.
Right. And that's why I say that the vaccine is probably the way to go, because you're right. The coronavirus is really having effects on a number of different systems. Circulatory system, you're seeing all these blood clotting effects. There's some reports of kidney trouble, and maybe even in some patients, some long-lasting effects that go on for months or maybe even the rest of their lives. So that's bad. It's bad because of the medical problems, and it's bad because it means that there's a lot of really complicated stuff going on there.
Yeah.
It could take us many, many years to unravel the real details of why all these bad things are happening. So the best thing that we should be able to come up with is something that keeps you from getting infected to start with. And that's the vaccine. Because if we if we end up having to chase down all these effects on different organ systems, that's not going to be good. That could take us quite a while.
So I've also seen reports about the spike in antibiotic uptake due to the coronavirus. So initially, I thought, you know, it was people self-medicating, but it seems that some doctors are prescribing the antibiotics for these secondary infections. So
Right.
do you think that maybe with this surge of antibiotic prescriptions, we're heading towards, or we risk moving towards another pandemic because of antibiotic resistance?
Yeah, I hope not. But I know what you're asking about, because that's the problem with antibiotics, is that the more of them you give, the more chances you give the bacteria to become resistant to them. It's a big problem with viruses as well. These things reproduce so quickly. They turn around in a matter of sometimes just an hour or so, and they've split in two and made more. Viruses, within a few hours, you've got thousands of times more virus going. So if any of these things hit on a mutation by accident, if they hit on a mutation that makes them get around the drug you're giving, that can spread really quickly. And that's what you see, is that you're giving them what I guess a biologist would call selection pressure.
Yeah.
You're telling them, get around this or die. And the ones that get around it don't die, and they take over, because now the field is wide open to them. All the competition that could be killed by the antibiotic or by the antiviral is gone, and now they're ready to go. So that is the problem. And antibiotic resistance for bacteria is, of course, a long-standing problem. And anything that revs up the use of antibiotics, you're right. That's something to be worried about. Another reason to hope that we get some sort of preventative, to where we don't keep handing these things out.
Yeah. And then given what's happening outside of, you know, the COVID-19 pandemic, as you're aware, there's been a lot of conversation about anti-Blackness, and especially in medicine and the drug discovery industry. Historically, there's a really bad relationship with the Black community. So what has the industry done, or how has it responded to some of these conversations?
Oh, yeah. Yeah. No, isn't it something that we've got this boiling over at the same time as a pandemic?
Yeah.
And in the US, at the same time as a presidential election. 2020 is a new one. But yeah, you're right. The relations between drug research and the Black community have historically been awful, and awful for some pretty damned good reasons. The original sin there was the Tuskegee syphilis experiment.
Yes.
That, I cannot tell you the number of times that people since then had wished that that never happened. Never happened, one, because it was an inhuman thing to do. Totally inhuman and wrong. And two, because it has poisoned the relations between drug research and medical research and the Black community ever since, as it should, because ever since then there's been this long-standing suspicion of people coming to do experiments on you, and I can't blame anyone for having that suspicion. It's awful. So yeah, the drug industry has realized for a long time that it's got a problem like this, but recent events have brought this up even more. And you can see from the statistics that the Black community is hit harder with the coronavirus. And I think a lot of that is unfortunately socioeconomic, because these are communities where the healthcare wasn't as good to start with. I don't know if there's something particular in the various ethnic groups that make different ones more susceptible. I have not seen anything like that. I think it's probably just standard of healthcare, which is bad in one way, but it also means it's something that you can address, and it's something that you can try to correct.
has there been any efforts to, for example, include especially these Black patients in either the monoclonal antibody development, or, to really look
Yes,
at the genomes and see if there's differences?
there has. And there are also efforts to make sure to recruit a lot of people into the clinical trials. But of course, we were just talking about the
Yeah.
suspicion between the Black community and drug research. This is one of the bad effects, is
Yeah.
that you would want to correct this by saying, okay, we've got to make sure to test these drugs in the Black community and make sure that these things work well. And then many people in the community are like, oh yeah, you're coming to test more drugs on us. So it's awful. But yeah, there is a concerted effort to try to recruit a good cohort of the population to make sure that we're not missing something, and to make sure that these drugs are going to be equally effective.
So, besides the US, what other markets are conducting clinical trials? I think China also has some.
Right.
Do you know of any in Africa or in Europe?
Yeah, that's a good point, because I believe that there are some trials. I believe that some of the trials that are going on are going to be recruiting in Africa, because here's the tricky part. You want to test these antibodies and vaccines in places where there are a lot of coronavirus patients. And that means that now, if you want to try to do it, say, if you wanted to test in Germany, well, you're out of luck, because Germany has done a good job containing their coronavirus. If you want to do it in, say, I don't know, South Korea, you're really out of luck. South Korea did a great job containing their epidemic. So, problem is, there are going to be too many places in the US where you're going to find people. We're trying to make sure we don't run out of coronavirus cases, God help us. But it means also that right now, South America
Mhm.
and the Middle East and Africa are the places that are climbing up now.
Yeah.
Brazil has a serious problem. Chile has a serious problem. Peru looks like it's getting into trouble. And South Africa,
Yes.
Nigeria, these are places that you're going to have to keep a close eye on. But if the epidemic is really getting a foothold there and really starting to take off, that's probably one of the places that you want to go and test your drug, because you're trying to give people a vaccine to prevent them from getting infected. So you want to go to some place where a lot of people are getting infected, and you can get the best numbers the most quickly that way.
Are you aware of any collaborations between universities or pharmaceutical companies here with universities or healthcare centers, or even drug discovery companies outside of the US, for example, in Africa, to kind of have this multi-efforts development?
Yeah, some of the infrastructure that's been in place for things like the Ebola epidemic and the and also the tropical disease, the subtropical disease eradication efforts. Some of that infrastructure is being brought in for this as well. So you have the contacts between the pharmaceutical companies and the multinational agencies, and the actually the institutes and organizations in Africa who are there and know the territory, they know the hospitals and the research centers, and they know how to recruit patients. So that's one good thing, is that that infrastructure is there because of these efforts. And you can turn around and say, okay, we're looking at this disease. Let's also contact you about this one. So I think we're going to see that infrastructure being used if the clinical trials move into some of the sub-Saharan countries.
Okay, going back to the anti-Blackness, I think one of the, so apart from the Tuskegee experiment,
Tuskegee, yeah.
there also has been this historical relationship with quote unquote Western countries
Yeah.
giving African countries, drugs such as Depo-Provera, or if something's taken off the market here, it can still be marketed at home.
Yep.
And I
Yep.
guess I'm also afraid that we're going to meet this the same distrust from our communities,
Yeah.
from the African communities. And I think in the industry, because I've worked there,
Right.
part of the problem is there are no black faces, right? So besides Ken Frazier,
Yeah.
so
Merck.
who
Yeah.
else is there? And just that lack of representation almost signals to people that either you don't care or there's no consideration of this
Yeah.
population.
Yeah. It's a problem, and of course that lack of representation you're talking about is, I hope, partly a historical artifact, because the people who have worked their way up into these management positions have been doing so for the last 30 years.
Mm-hmm.
And 30 years ago there were even fewer black faces, et cetera, et cetera. But you're right, there's a problem with the mistrust that you're talking about. And there have been instances from the generic companies in India and China
Yes.
where documents have come out where they have deliberately tried to unload substandard material into markets where they think they can get away with it. And Africa is one of those markets.
Yeah.
South America gets some of that too, but Africa is one of those markets. And that, of course, is unconscionable. That's not only wrong, but it's inhuman to do that. So I think that there's a big break in the pharmaceutical industry between the companies that are doing the research to try to find new drugs and the companies that just make the cheap generic drugs as cheaply as they can. And that is one of the big splits in the industry. If you're not in the industry, you don't necessarily see that. You think, okay, here's the drug industry. Here they are. They're all together. We're not all together. The companies that spend a huge amount of money on research and development are different from the ones that are trying to crank out generic drugs as quickly and cheaply as they can. We need both kinds. Generic drugs can be excellent. They are terrific, and they're very cheap because they've gone off patent.
Yes.
All of our expensive drugs are going to turn into generic drugs, and that's how it should be. You only get a limited monopoly, and then it becomes cheap. But you have to look out for the people who embrace that cheap and fast philosophy so much that they're willing to harm people. And I cannot tell you how much I support coming down on people like that. We have to keep in mind that people are not always very good. And if they decide that it's okay to harm others in the service of making a buck, whether it's the drug industry or anywhere else, you come down on them hard. So when I see a company fined for having crappy manufacturing practices in India or wherever, I'm like, good. I don't get all upset because they're coming after the drug industry. I'm like, good. Every company that does this should be hammered on.
Yeah.
As an example to everyone else not to do this. People in theory shouldn't even think of doing stuff like that, but they do. So when they do, they should get hammered on. I want them to lose their businesses. I want them to lose their livelihood. We can't have this.
Yeah, I think this is a good segue to discuss pricing and accessibility.
Sure.
So when these drugs, so part of this problem of the generic industry, I guess historically has been that the brand name or the companies that develop these
Right.
treatments, they start off with some exorbitant prices, and not everybody can afford it. And that's how the generic industry kind of came to life, and was also an effort by Western governments to provide medications for especially the global South to have access to some of these medications. So I heard Gilead was promising to sell remdesivir at cost. Do you think everyone else is going to do that, or?
I think so. To be honest, I think so, because, for example, Gilead has said that. Johnson and Johnson is one of the big vaccine players. They have already said that they are doing their vaccine work on a not-for-profit basis. They're not trying to make money off of it. I think, and Regeneron CEO has said the same thing. They said, look, we cannot come in and look like we're swooping up this big windfall profit because of the coronavirus epidemic. I think everyone in the industry realizes that there's actually, to be honest, there's a lot of good public relations in being a drug company that's attacking this virus and
Yeah.
maybe being one that can come up with a real treatment or cure. We should be happy with that. The industry needs a better reputation, and we need to remind people of what we can do and how we're able to help. Let's do that. And I think that so far, all the companies I've seen seem to be well aware of how awful it will look
Yeah.
to come in and make a great big pile of money off this wonderful pandemic. That would be a disaster.
Yeah.
So yeah, I think that's what we're going to see, is that it's going to be a combination of companies working on a not-for-profit, of governments putting up money, of private foundations like Gates and others putting up money. One way or another, we've got to get these treatments around the world, and we're not going to be able to charge everyone every dime they have to do it. It's in the best interest of the companies, the countries, and of the entire human race to do
Yeah.
this. And I think we're actually going to do it.
Okay. So I'm going to switch gears to talk about science communication.
Okay.
So how important do you think science communication has been in managing this pandemic?
Yeah, you could be really optimistic or really pessimistic, depending on what kind of mood you were in when you woke up. Because the signal to noise, as a scientist would say, the signal to noise during this epidemic has been terrible.
Yeah.
Just terrible. But that's kind of what you would expect, because it hit kind of suddenly. A lot of people don't understand about viruses or drug discovery epidemiology, and people are very worked up, as they should be, and emotional. So it gives a chance for a lot of information and misinformation to spread, and the misinformation spreads faster.
Yes.
So, like you were saying earlier, it is important for scientists to speak up and try to get good information out there. That's what I've been trying to do on the blog.
Yeah.
And it's tough, though, because misinformation spreads more quickly because it's more tailored to what people want to hear. If you spread some rumor that there's this amazing cure that the companies are keeping from you,
Yeah.
that is exciting. That's the sort of thing you feel like hitting forward to and sending to your whole Facebook group and calling people up about, as opposed to having some dude from the drug industry come along and say, gosh, it's really hard and it's really complicated. I don't know how long this is going to take. That's not very exciting.
No, it's not. And what do you think the role of Twitter or digital media has been, especially in combating this misinformation or doing some of the science communication work?
Yeah. It's got, unfortunately, two roles. One, it is the finest way to spread misinformation that the human race has ever seen. But at the same time, it allows you to respond to it quickly. Sometimes it works well and sometimes it doesn't. There's an old saying, I think it's attributed to Jonathan Swift, but at any rate, it goes back to like the early 1600s, that says that a rumor is halfway around the world while the truth is still lacing up its shoes. And
Yeah.
you see that a lot.
Yeah.
Problem is, people believe what they want to believe. And, well, we were talking about hydroxychloroquine earlier. As time has gone on and we've gotten more controlled clinical data, hydroxychloroquine looks less and less like a useful drug. But the people who truly believe this are never, ever going to be convinced by that.
They're not, yeah.
Never. And, you know, here's the saying that I was thinking about earlier. It's not from me, but I'm fond of quoting it. You can't use reason to argue someone out of a position that they didn't get to by reason. It's the wrong tool for the job. It's like trying to pull out screws with a hammer. And the hydroxychloroquine people, for example, just to pick one example, believe this because they want a cure. They also get a lot of satisfaction from having someone that they may like Donald Trump, tell them that this is the cure. And it also, frankly, it pisses off people that they hate to have Donald Trump say this is a cure. So what's not to like? You've got a cure for the thing. The person you like is with you, and the people you hate are angry about it. It's wonderful.
Those snobby scientists.
Yeah, so they're not going to give that up. They're 10 years from now, when I hope that we have all started to forget about 2020 and the coronavirus, 10 years from now, there are still going to be people on Twitter talking about the huge conspiracy that kept hydroxychloroquine away.
Yeah. Well, Derek, thank you so much for agreeing to be on my platform. I think my audience is really going to appreciate it. This has been really informative. So we're going to do a lightning round, and you just say the first thing that comes to mind.
Oh, no.
So what activity are you doing now during the pandemic, and that has been an addition to your everyday life?
Let me see. I've been working from home, of course, which is okay because I don't do that much work on the bench, so I can do a lot of what I do at home, which is good. But I also try to make time, honestly, to get outdoors. I go out and I work in the garden, or my wife and I deliberately try to go out to some local forest or park or nature reserve or something and just get outdoors a little bit. It's a good reminder that not everything is people screaming at each other on Twitter about the coronavirus.
And in a perfect post-pandemic world, what is the first place you will go to, or the first activity that you will do?
Oh my gosh. Well, my wife and I actually had a vacation planned back in March, late March, and we had to cancel it just as this thing was getting together. So she's turned around to me and she said, you know what, as soon as this is all over, we're going to travel. Yeah. So I got a couple of restaurants locally that I'm looking forward to sitting down in and eating again, but I will very much enjoy actually just driving or flying to some place and enjoying myself in new surroundings if I can. We didn't realize how much of a luxury that was.
Yeah. And what's one thing that has come out of the pandemic that you hope you keep?
You know, the working from home stuff has its irritations, but I think it's actually, a lot of people have realized that a fair amount of their work can be done that way. I hope we do keep some of that. It's not so much fun when you're forced to do it, but there are days in my normal working life where I really had no reason to go in to the building where I work. So I hope it becomes more acceptable to do it like that. I think it'll actually improve people's work and life balance.
Okay, well, thank you. Where can our listeners find you on social media or by email?
Absolutely. Well, on Twitter, I'm at Derek Lowe, DerekLowe. And if you search my name on Google, you will get a former Red Sox baseball pitcher and you will get me. Those are the only two Derek Lowes that show up on the first page of Google. And that will send you directly to my website, which is called In the Pipeline. And my email address is right on the website. So I'm actually pretty easy to get a hold of. And I have no problem with people contacting me with questions or ideas or things to write about. No problem at all.
Okay, thank you, Derek.